All right, so this is going to be about translation. Translation's a little bit complicated, so we will try to get through this as painlessly as possible. And the first thing you need to understand is what is mRNA. We've covered this last section, but just in case you forgot, mRNA was literally made from one gene in your DNA. So each gene in your DNA codes for a protein. This particular piece of mRNA is a copy of that gene. You did it by matching, right? So the DNA that this matched to, A went with T, U went with A, right? Because remember, T and U were exchanged in RNA. RNA uses U instead of T. G, so there was a C in the DNA. C, there was a G. UA, UA, right? So we could predict what the DNA was that this messenger RNA was made from. And the messenger RNA then carries the message, the information from the DNA. And if you look here, it's giving you a hint as to how you read messenger RNA, how your cells read messenger RNA. And it reads it in triplets, in threes, three nucleotides. So if you look here, AUG, AUG is a codon, right? It's 1, 2, 3 letters. So it's a codon, and that's how you would read that first three. And then CUU is another codon. So this is how your mRNA is read. It's read in triplets, and it's read from a excuse me. It's read from a start, which is one of the starts that we use is AUG. It's read from a start, and then every three after that start. So AUG again is the start, and there's three stop codons. There's three ways to stop, and I'm going to show you a chart here. This chart will help you see what each codon does. Now this chart is complex. There's no ifs, ands, or buts about it. But if we break it down piece by piece, it won't be a big deal. So let's look at AUG, right? That was the first one we saw. So we're going to look at AUG. And see here it says first base. Here it says second base. And here it says third base. So this is not for baseball. This is the triplet, the first letter, the second letter, and the third letter. So the first letter for AUG is A. So UCA, right? The second letter in AUG is U. So the second letter is U. So where A and U combine, that's this box. And then the third base, UCAG. See how each of these boxes has 4 letters, right? So those represent each nucleotide. So AUG, and if I look here, AUG, and it tells me methionine, met. Now methionine is an amino acid, okay? And that amino acid is the start, also the start codon. That's the one that starts it. So the example they're giving here is CAU. So if you have the codon CAU, okay, you go to the first base, here's C. You go to the second base, here's A, C, A, and then U. And you're right here, right? And that's histidine or his. And by the way, had you had found CAC, you would have also gotten histidine or his. And that happens. There is some duplicity here where you get more than one way to get the same uh, amino acid. For example, if you looked at these, CGU, CGC, CGA, and CGG, all give you arginine, Arg. And these are all amino acids. Let's talk about some of the things logistically here, okay? What do you need to know from this? Do you have to memorize this chart? No. I do not want you to memorize this chart. I do not want you to memorize what each of these 3 letters means, like where it corresponds to. Nor do I want you to know the names of all of these proteins here. But if I gave you 3 letters, you should be able to tell me which protein, which amino acid it goes to. So if I gave you GGG, right? You'd be like, okay, G, the second letter G, G is glycine because it's G first base, G second base, G third base, glycine. So you should be able to use this chart to figure something out, but not necessarily memorize the chart to figure something out. See the difference? So, and that's how you would use this chart to get through this information. And you know what? We will continue our discussion about 8.5 after this.